Hi everyone, welcome to AICE Psychology. I will be your guide today through Dement and Kleitman. So stay tuned and we will learn about REM and dreaming. Okay, so let's get started on the background information. So just like any other study, you need to understand the background psychology behind this, or even what was happening in the world at the time, just to understand some of the groundbreaking information, scientific information that we discovered in this study. So we are in 1957, and that is the year that this study was published. It wasn't the year that it was conducted, because Dement had a really hard time publishing this study. I believe it took about 9 times before he was able to publish this study for the public to read. And that usually only happens with groundbreaking information, or, you know, if someone believes that someone's idea is kind of off the wall. So dreams was thought to be something that was mystical and mythical, and we didn't really study it too much because it was really subjective. What someone dreamt about was not the same as what other people dreamt about. And I'm sure you've had many people say, oh, I had this awesome dream, and they start to tell their dream and it was a really dull story. Because we have so many visuals that happen, again, that are subjective, and we are asleep when they occur. So we have not had any prior research or evidence that explained that this phenomenon was real, that it was objective, that everyone dreamt, everyone slept the same way, or even that there was a sleep cycle. So it's going to be important for you to know that there are 4 stages of sleep. You don't have to go too in depth in studying them, just understanding the cycle of sleep, maybe the mean length of a REM cycle, and so forth. So there are 4 stages. Look in your book. I would recommend that instead of going online, because there could be 5, 6 different stages depending on whoever made the graph and how they made it. But there are 4 stages that you need to know, and let's go through them really quickly. So to start, when you're awake, we have what's called beta wavelengths, and they're very rapid waves that occur because we have a lot of activity that's going on. Our brain is quite active. We are thinking many things. We have a lot of emotions going on. So that's beta waves. So right before you get to sleep, you know, your head just touches the pillow and you're calming down, your breathing slowing down. Those beta waves slowly turn into alpha waves. And from there, they go into theta, so they're just slowing down and slowing down and slowing down, till basically we become completely asleep, and we are in what's called the heaviest sleep stage. It's the delta wavelengths, and this is where growth hormones are released, so we find that a lot of young children sleep really heavily in this delta wavelength period. I know that I can pick my kids up and they're, you know, completely limp, and I could throw them up on their bed and they don't budge or wake up. So I can pretty much tell that they're in that delta wavelength period of their sleep in that moment. And then from delta, we have our REM. But you should understand that it doesn't go stages 1, 2, 3, 4 REM, because from delta wavelengths, we have to get ourselves back out of that sleep. So we're in this really deep sleep, so we have to kind of wake ourselves back up, which means more activity in our brain. So we go from our awake of beta to alpha to theta in this deep, deep delta. And then our body starts to wake us up a little bit. We go into theta and then back into alpha. And instead of our body, you know, putting our brain activity back into an awake state of mind, we go into what is called REM, rapid eye movement. And in this stage, our brain mimics our awake mind. So we have a lot of activity that's going on. We are seeing a lot of images. The only difference is that our body is not moving in REM. Okay, so our brain goes from beta, alpha, theta, delta, back to theta, alpha, and then into REM. So that whole cycle that I just stated takes approximately 90 minutes to complete. So it takes about 90 minutes for you to get into a sleep cycle. And we'll see that one of the results from the study stated that there were no participants that entered REM directly after falling asleep. Okay, so 90 minutes was the average sleep cycle for someone. About 20 minutes was the average time for REM. But what the research showed was that over that long period of sleeping, we'll just say 8 hours if you're lucky enough to get 8 hours of sleep uninterrupted, that each time that you enter REM, it gets longer and longer and longer and longer. So anywhere from a 5-minute period in the beginning to maybe at the very end of your sleep cycle, maybe you're having a 60-minute dream. Okay, remember I said average. So we're taking that was the average time, was approximately 20 minutes. All right, so a few things that you should understand before diving into the study is REM, REM, rapid eye movement, and NREM, non-REM. So when we're talking about non-REM, we're basically talking about every stage in the sleep cycle except for REM. Okay? So with that being said, let's talk about what you need to know from every study for paper 1. Okay? So we have our aims or hypothesis. We have our sample, not only who they were, but how the sample was collected. You should know your independent variable. You should know your methodology, your methods, and you should know your design of the study. You should also be familiar with the controls, the procedure, any ethics that occurred, good or bad, and the findings and conclusion of the study. Okay, so let's get started. So in Dement and Kleitman, we have 4 aims. 3 are true aims, and then 1 was like a sub question that was asked during the experiment that we kind of held on to as an aim. Okay, so let's start with aim 1. In aim 1, we are looking for a relationship between REM and dreaming. So we are looking for yes or no answers when we wake our participants up from a REM sleep or from a non-REM sleep. So if you were the participant and you went to bed, we're going to wake you up and we're just going to ask you one question. Were you dreaming? And you just answer yes or no, and then you go back to sleep. We didn't tell the participants if they were dreaming or not, or if they were in REM or not. That's going to skew our data, domain characteristics, big time. Okay, so we're going to get quantitative data out of that. A yes or a no can be like a check mark system. So we're going to say it's quantitative, and it's objective because we are using our EEG machine, which is scientific equipment, and that's always going to elicit objective behavior. Okay, so in aim 2, we are looking to see if we dream in real time. We really want to know that question, is a dream that feels like an hour in our head, is that really an hour in real time? Or if we have a 5-second dream, is that really 5 seconds in real time? So how we figured this one out was we, again, waited till our EEG machine either told us that our participants were in REM or not. Once they were in REM, we let them dream for, let's just say, approximately 5 minutes. If we let them dream for 5 minutes, then we woke them up and we asked them, how long do you believe you were dreaming? If they said approximately 5 minutes, then we have some type of relationship there. And then we would ask other participants, did you wake up 5, 15 minutes? The reason we went back and forth between 5 and 15 minutes was to kind of throw off participants. Again, domain characteristics, we don't want them to be there. We don't want participants to guess that 5 minutes because we're not going to know if they're guessing or if they're being genuine in their answer. All right. Again, that's going to elicit some objective behavior. We have our EEG machine telling us that they are in REM. And then they're going to tell us a time of, 5 or 15 minutes. Honestly, it's really close. So we're at least hoping that they're going to say that when they dreamt 15 minutes, that it was longer than that 5-minute duration. Now for aim 3, we are looking to see if there is a relationship between REM, our rapid eye movement, and the content of our dream. So what are we seeing in our minds when we're dreaming, and what are our eyes actually doing? Because we know they move, but why are they moving? So in this aim, we begin to see, well, our first qualitative data, written words, something that is being paraphrased, which is their dream. And we still have our EEG machine that's telling us which way the eyes are moving, but we have some subjective interpretations of the dream. So again, we're looking at to see if there is a relationship between the way someone's eyes moved and their dream. So for instance, if someone's eyes moved left to right, horizontal, we saw that they dreamt about something that required horizontal movement, like walking through a field. If we had a participant's eyes that moved vertical, up and down, then we tended to see dreams like rock climbing, dreams that involved someone moving their head or looking up and down. We did have another category where it was both up and down and back and forth. And we saw a lot of dreams that had to do with like up and down, side to side movement, like maybe basketball. Okay, so it's important to know that in that aim, we could consider it to be subjective, and we have qualitative data. So it's not objective like we're hoping for with the first two aims, but it would still be a fun correlation to find out at the end of the day, if the numbers were right. So when I say objective, I mean that the person cannot argue against it. There's no gray areas. If I say 2 plus 2 is 4, that's objective. But if I'm telling a researcher about my dream and they are interpreting my dream as vertical or horizontal themselves and placing it in their own category, then technically that is being subjective. Okay, so remember that. Now, the fourth aim has to do with the duration of REM sleep with the number of words in the narrative that's being reported. So basically, we were just monitoring to see how many words on average people use to describe dreams. And if we found that someone wasn't descriptive enough, then we would just consider that a non-dream. We're basically hoping to see the more instances of dreams and detailed dreams occur while people are in REM. And that was just subsequent information. We didn't stop participants to test that aim specifically. Okay, so the method of the study, we have a natural lab experiment. The reason that it is natural because our participants naturally sleep. Sleeping is something that we naturally do, so we are going to consider it a natural experiment. We're also going to consider it a laboratory experiment because it is foreign place to our participants. If it was someplace that they naturally slept, like their own bed, then it would definitely be a field experiment. But because it's in a lab setting, or even because it's in a bed that they normally don't sleep in, we call it a lab experiment. Okay, so the variables in this study are not just cut and dry. We have a variable for each one of the aims, an independent variable and a dependent variable. So for aim 1, the independent variable was REM and non-REM. Okay, and we are waking participants up, and the dependent variable was dream recall. So we are looking at the number of dreams that were recalled in REM and non-REM. In aim number 2, we are looking at the duration of time. So our independent variable is the 5 minutes and the 15 minutes, and our dependent variable was the correct length of dream. So we have repeated measures for the first aim and the second aim because our participants are being measured on both sides of the independent variable. So in aim 3, our independent variable was the direction of eye movement, horizontal or vertical, and our dependent variable was that subjective story or narrative that was told by our participant as whatever the dream was, whatever the content of the dream was. And for the fourth aim, we don't really have an independent variable, but we were looking to see if the number of words used to describe the dream correlated with the amount of time we let our participants dream. So the design of the study is a repeated measures design. What that means is our participants are being measured on both sides of the independent variable. So let's just take aim 1, REM and non-REM. If you were woken up in non-REM and measured, and then you were woken up again in REM and measured, then that is a repeated measures design. Okay, we do know that participants did come back to get measured multiple times, but this study is a little bit different than others because we're dealing with sleep. And I don't know about you, I know some people are a little different and they can sleep for long periods of time. But if you are sleeping multiple 8-hour periods in 24 hours, then some data is going to get skewed because that's not something that we normally do. Okay, so let's talk about the sample of the study. Really important here. We originally had 9 participants. There were only 2 females, the rest were males in the study, but it was kind of interesting what happened with 4 participants. They did exercise their right to withdraw, and they were only tested maybe 1 or 2 nights. So we just used them as supplemental data to kind of prove anything that we saw from the 5 participants that made it all the way through the study. So it is a self-selected, which means they volunteered, but we don't know much about how they knew about the study. We do know that the study was done at the University of Chicago, but we don't know how participants knew about the study. So the fact that they volunteered, they had to have known that they were sleeping and maybe that they were going to get woken up and asked a few questions. But other than that, we don't know much about our demographics here. Just keep in mind that our population is really small, but we did use repeated measures, and a good thing about repeated measures is that you don't need a large population. So I still think Dement and Kleitman are okay on this, but remember, we did only study 1 female out of those who stayed in the study for the long term. So I'm not exactly sure that you can say that the study is going to be generalizable to all males and all females. Okay, so let's talk about the apparatus. So we are in a lab of some sort. We have our EEG machine, the electrodes that are connected to our heads, that's part of the apparatus. You can also consider the tape recorder and the doorbell to be part of the study because they were used by all of the participants. So let's talk about the controls now. The controls of the experiment were important because they're going to help us to improve the reliability or the validity of the study, depending on what we're controlling for. So an example would be that we told all of our participants to report to the lab at their normal bedtime, but we told them to avoid any caffeine or alcohol prior to the experiment. They could drink and eat anything else they wanted, but had to avoid caffeine and alcohol. That is a good control because we are ruling out caffeine and alcohol as any reasons why someone might dream or sleep for so long or dream certain content or have certain brainwave activity. I know for a fact that if I drink a cup of coffee too late at night, it really hinders my ability to fall asleep and stay asleep for the whole night. So that is a good variable to have. Another control was that all participants were woken up with a doorbell. We wanted to keep that consistent. Now, that lowers our ecological validity a little bit because people aren't normally woken up with a doorbell, but at least it was consistent, and we can't say that maybe one person, for example, if we had to yell at people to wake them up or just, "Hi, honey, wake up, can you wake up?" I mean, that might be different for every person. You know, everyone sleeps differently. We have heavy sleepers, light sleepers, so the way that you woke up, it's important, but instead of having a person do it and probably risk being biased in any way, we just had a doorbell wake participants up. Okay, so this is part of the experiment where you have to put yourself in the shoes of the participant. And I'm telling you, this is the only way that you're really, really going to understand and be able to envelop yourself in the study. We have 12 studies that you have to remember, and knowing the detail and being able to play them through your head, it is really important. And visual memory is a good one in the study because we don't use a lot of it in our textbook. There's not a lot of pictures in our textbook. So try to imagine this happening. Try to imagine you being the participant, okay? So it's an average day, and you know that later on that you have to go to the university sleep lab because you're going to be part of the study. And all you know is you can eat and drink anything you want except caffeine and alcohol, and you're going to show up there at 7 because that's the normal time that you normally go to bed, okay? So you enter the sleep lab, and you are given electrodes, and they're put on your head, and this is hooked up to the EEG machine, so we're going to be able to see brainwave activity through the machine with these electrodes, and everyone has them, so that's another control of the study. So, you know, I don't know about you, but I may not be able to sleep with these little stickers on my head and wires in my face, so what they did was they just put all the wires back in like a ponytail, so to speak, I think a little higher on the head, so that nobody would be disrupted by those wires when they're sleeping. And if anyone is, at least everyone had the same experience, and it wasn't just one person out of, you know, the 5 that were left. So you're in the study and you go to sleep in a nice, quiet, dark room, and you've been sleeping for a couple hours, and ding dong, you hear a bell. You're asked a question. Were you dreaming? What your job is to do is to go over to the nightstand next to you, and there's a little recorder. You hit the intercom, a recorder. You hit it and you speak into the intercom. You're not talking to the researcher. You're just kind of recording your answer, and then you go right back to sleep. So you may not know the aims that they're asking you. You may not know why they're asking you certain questions, if they're going to repeat a question again, if they're going to jump to another question. But your experience as the researcher is kind of detailed compared to what our participant's doing. Our participant is sleeping, being woken up, asked a question. They're either saying yes or no, or they're providing detail, and then they're going back to sleep. It's the job of the researcher to be watching to see when the EEG machine is telling them when you're in REM, when you're not in REM, what questions are we going to ask, which question did we already ask. We have to make sure that we aren't repetitive, so, you know, we don't elicit domain characteristics. All of those things are really important. Let's talk about some of the results. So we found that every participant dreamt every single night they slept. So if you are a person that says, I don't dream, you dream. You just might not remember it. Because like I had said before, your dreams get longer and longer and longer as the night goes on. So if you're not sleeping well, if you're not sleeping long enough, you're probably going to only have small instances of dreams. So it makes perfect sense that you might not remember, but remember that everybody does dream. We also found out that on average, a dream lasts approximately 20 minutes. And we also found that an entire cycle takes about 90 minutes. So to reach, I think it was 92 minutes on average. So to go from REM to another REM is approximately 92 minutes. And although a lot of this information might seem common knowledge now, remember this was 1957. So some other pieces of information that we found, that participants never entered REM upon sleep. They were always in non-REM right when they fell asleep. And when participants were woken up in non-REM, they went right back to sleep in non-REM. When participants were woken up in REM, they fell right back to sleep in REM. Now for our conclusion. Dreams are more likely to occur in REM sleep. Dreams appear to happen in real time. And dream content seems to correspond to the direction in which our eyes move. There are some areas in the evaluation that you should key into for paper 1. It's suggested by the Cambridge book. Talk about the scientific equipment, because the use of scientific equipment can improve the reliability of the study. We should talk about the controls, because controlling for variables is highly desirable, but it also can end up lowering the ecological validity like we saw in this study. And generalization. So can the findings of this study be generalized to everyone? Do we all have REM and non-REM? The answer is definitely yes. So it's tricky. We don't have a lot of males and females to go off of. It's a small population. But again, it's all in how you word your evaluation. All right, everyone. So there you have it. We went over the aims of Dement and Kleitman, the sample, the methodology, the designs. We talked about the independent, the dependent variable, the apparatus of the study, and the controls. So you need to focus mainly on those results and conclusions, because evaluating the study is going to be really important, which we will do in another video. So stay tuned, watch other videos, learn apsychology, pass that test, and if you have any questions, you can send me a message at apsychology9990@gmail.com. You can find me on Instagram at apsychology. And For those of you who subscribe, send me a message and I will hand you the code to my Google Classroom that has all 12 studies, all the evaluations. It has all of our vocabulary and our strengths and weaknesses. So you will get all the material that you need to pass your exam at the end of the year. Subscribe and like my videos. All right, have a great day and I'll see you next time.